Hello guys, and our topic for today is the renal tubular processing. It's the part 1. We will discuss a series of lectures and we'll cover this topic. So we will discuss here that what are the processes and mechanisms occur in the renal tubules for the reabsorption of the filtrate. Because as you know that the basic steps in the urine formation involve the filtration we have discussed in the previous lectures, and the second is the tubular secretion. It can be passive or active, and the third is the tubular reabsorption. And tubular reabsorption is of very high importance because 99% of the glomerular filtrate is reabsorbed and only 1% urine is formed in the whole day. We'll discuss it now. So as you know that basic steps in the urine formation, first is the formation of the glomerular filtrate. It means that from the afferent arteriole the glomerular capillaries arises, and from there the filtration process is going on, and everything in the plasma is filtered. The water, glucose, ions includes sodium, chloride, potassium, magnesium. All of that stuff is filtered except of the plasma proteins because they are of high molecular weight and they contain specific charges, negative. For example, albumins have negative charge, and the filtration barrier also has negative charge, so they repel each other. And the substances that are bound to the plasma proteins are not also filtered. The second step is the absorption, basically reabsorption. And the absorption can occur through the cells. It means that transcellular absorption. Okay, it means that if the absorption is occurring through the cell, okay, it's the transcellular absorption. And if the absorption is occurring in between the cells, so it's the paracellular reabsorption. Okay, so it's very important to remember these two terms. The third step in the urine formation is the tubular secretion. It means that most of the substances, for example, drugs, potassium, etc., we'll discuss them later. The tubular secretion, the substances are secreted actively from the tubules into the nephrons, and these three steps collectively form our final urine. So our equation for the urine formation is urine excretion is equal to the filtration plus the tubular secretion and minus tubular reabsorption. It means that the stuff that has been reabsorbed is minus from the stuff that has been secreted or present in the lumen. The second important thing here worth mentioning is the basic principles of tubular reabsorption. Our topic is the tubular reabsorption. Collectively, both kidneys have 2.4 million nephrons. It means that a lot of nephrons are present in both kidneys. But from now on, we will discuss only one nephron, and that process occurring in one nephron will occur in all the other 2.4 million nephrons. As you know that the glomerular filtration rate in 1 minute is 120 ml per minute. Okay, it is the filtrate. The filtrate formed in 1 minute is 120 ml. So in this diagram you can see that the filtered stuff that appear in the Bowman space is 120 ml per minute, and some persons or some variable that we can estimate is 125 ml per minute. Okay, 120 to 125 ml per minute of the filtrate is formed in 1 minute. But if we want to calculate the GFR in 24 hours, so what should we do? 24 hours contain 1440 minutes. Okay, our whole day contains 1440 minutes. It means that if we multiply this number with 120, we'll get the estimate amount of GFR that is produced during the 24 hours or the during the day. It's approximately 17 and 18 liters. 17 to 18 liters per day of the filtrate is formed. You can imagine the our filtration power of nephrons. It means that our body has 5 liters of blood. but it is passed through the nephrons again and again until the 18 and 17 to 18 liters of filtrate is formed in 24 hours. And this is filtrate, it is not the urine. Our estimate amount of urine for the whole day is 1.5 to 3.5 liters. Okay, we will take this number because we are considering just an estimate amount. So all of that 19 liters is reabsorbed. It means that all of that 17 liters, sorry, is reabsorbed. 99% of that, 99% of the filtrate is reabsorbed. So reabsorption has its own significance when it comes to the nephron physiology, right? And calculating the amount of urine that is produced in one day, we can calculate it. 120 ml of filtrate is formed in 1 minute, okay, and 119 is reabsorbed, and only 1 ml is appearing in the urine in 1 minute. So how many minutes are present in one day? 1440. So 1 multiplied by 1440, estimated amount of urine that is produced during the day is 1440 ml per day. But if we convert milliliter to liters, to 1.5 liters per day of urine is produced. Less than that will come into the term of oligouria, and more than 3.5 will come into the term known as polyuria. But the thing here I want to put in your minds that reabsorption mechanism is very powerful, and it's of great importance, great significance when it comes to the nephron physiology. So coming towards our main discussion for today is this diagram, and I will explain you this diagram in a minute. So first of all, we will see that how the reabsorption of sodium, glucose, and amino acid will occur in the nephron. So cells reabsorb sodium along with the water, chloride, glucose, amino acids. All of this stuff is reabsorbed into the proximal convoluted tubule cells. We have drawn two proximal convoluted tubule cells. These are only two, and a lot of cells are present here on the both side of the lumen. So 65% of the sodium is reabsorbed and 65% of the water is reabsorbed. 100% of glucose is reabsorbed because we do not find glucose in the loop of Henle, because the glucose has already been reabsorbed 100% in the PCT, and 100% of amino acids are reabsorbed in the proximal convoluted tubules. There are two membranes present in these cells. First is this membrane, the luminal membrane, okay, this one, and I am highlighting in yellow. It's the luminal membrane which is present towards the lumen, and the second membrane is the basolateral membrane because it is present towards the base and the lateral side. These are the lateral sides of the cell, and this is the base of the cell. So this is the basolateral membrane, and here is the luminal membrane. Luminal membrane is also known as the brush border because a lot of like brush present here, and the function of these villi, or we can say brush border, is to increase the surface area for reabsorption, and 20 times it has increased the surface area for the reabsorption. And the second term, I want you to know that tight junctions are present in between the cells. The proximal convoluted tubule cells are held together by these tight junctions that are present in between the cells. Okay, and the function of these tight junctions is not only the junctioning between the two cells, but it also allows the proteins that are present in the luminal side and the basolateral side to function in a proper way. It means that the protein present in the basolateral membrane cannot move towards the luminal side, and the protein present on the luminal membrane cannot move towards the basolateral side. So it keeps their function and their structures in a separate fashion. Okay, let's discuss about the luminal membrane, because in the luminal side there are important channels, and also on the basolateral side they have important trans proteins or transmembrane proteins. We can say important channels are present there. First of all, we'll discuss about the channels present on the basolateral membrane. You can see here in blue there are a lot of blue colored channels, or we can say they are pumps. Okay, they're present on the basolateral membrane. These are known as the sodium potassium ATPases. Okay, and the function of the sodium potassium ATPases is to move sodium out and potassium in. They move 3 sodium out and they move 2 potassium into the cell against their concentration gradient. It means that um, what does it mean? It means that the cell is already low. The cell is already high in potassium, okay, but it is constantly pulling the potassium in, and the extracellular fluid which is mentioned here is already high in sodium, so it pumps the sodium out and potassium in against their concentration gradient. When something is pushed against the concentration gradient, it will require ATP, it will require energy. This kind of transport that pushes the substance, or that transport one substance from its low concentration to high concentration, okay, it is known as the active transport. There are two types of active transport. First is the primary and the second is the secondary. Primary active transport means that the energy or the ATP is utilized at the specific point. It means that ATP will be utilized here, and this ATP will transport sodium out and potassium into the cell. And what does it cause? It causes the cell to become electronegative. How? Because a lot of cations are moving out than the cations coming in. 3 sodium are moving out and 2 potassium are coming in. In addition to this process, potassium also leak through the leaky channels. As you can see, the potassium is exiting out of the cell, and these two processes combine make the cell even more electronegative. The cell reaches the gradient of minus 70 millivolt, okay, and this electronegativity is very important. I will show you how. First of all, we will discuss that the filtrate is coming from the glomerular capillaries into the Bowman space, and here the concentration of sodium is very high because all of the stuff, sodium, water, chloride, amino acid, glucose, all of that stuff has come into the Bowman space as a filtrate. So let's talk about sodium first. Okay, sodium concentration here in the lumen is high as compared to the cells. Okay, the sodium concentration here is high. So there are special proteins present on the luminal membrane. These are known as the sodium transporters, and they transport sodium by the facilitated diffusion into the cell, because in this condition the sodium will move from higher concentration to low concentration. The sodium inside the cell is low and sodium in the lumen is high, so the pump will just bind the sodium, and it will reverse its direction and sodium will come in by the diffusion process. But why it's called facilitated diffusion? Because the proteins are being involved. The transport of substance from higher concentration, which is in the lumen, to the lower concentration, which is in the cell, requires diffusion process, and it also requiring the channel proteins, so it's known as the facilitated diffusion. And the sodium has come here. How this sodium move from the cell into the interstitium? Of course, by the sodium potassium ATPases. We have discussed that, okay? By the sodium potassium ATPases, the sodium will move out into the interstitium, and from the interstitium, it will move into the blood. The next thing I want you to know that along with the sodium can carry other things. For example, glucose, it can carry amino acids because the transporter which is transporting the sodium has two pockets. One can bind to the sodium and other is empty. It can bind either to the glucose or it can bind either to the amino acid. We'll discuss them one by one. Considering the glucose is bind to the one pocket of that co-transport. But one thing here worth mentioning is that is the glucose concentration here is high? Yes, it's high, but all of the glucose has been transported into the cell, and at the end of this proximal convoluted tubule there is no glucose, there is zero glucose. So it means that the concentration of the glucose inside the cell is high than the lumen. So how this glucose is going to move from lumen to the cell? It will move by the active transport. It is very important because all of the glucose has been reabsorbed. Even the last bit of glucose is also moved from lumen to the cell. So uh, in the last, the glucose concentration inside the cell rises and glucose concentration in the lumen will decrease. We will have to move the substance from low concentration to high concentration. It will require active transport. But is there any energy involving here? No. There is no energy involving here, so it's not the primary active transport. It is an other type of transport known as the secondary active transport, because the ATP is not being used here directly. It has come from another source, from the sodium potassium ATPases, because they have caused the decreased of the sodium concentration inside the cell, so that the sodium can move from lumen into the cell, and along with that it can carry glucose. Same thing applies with the amino acids. Amino acids are also co-transported with the sodium, and their concentration in the lumen decreases and their concentration inside the cell is high, so they also transported by the secondary active transport. It is very important to know that glucose and amino acids on the luminal side, they are transported by the secondary active transport along with the sodium, and on the basolateral side they just move out, they just diffuse out of the cell. So they will require a special type of proteins here also. So it's a diffusion, but glucose and amino acids cannot move through the plasma membranes. They are not lipid soluble. They will require a protein channel, and this is known as the diffusion. But as it is involving a protein or a carrier protein or channel protein, it will called as the facilitated diffusion. Okay, I hope you understand this, that let's revise all of that stuff that we have discussed. First of all, the first thing is first that sodium potassium ATPases will pump the sodium out, 3 sodium out and 2 potassium into the cell. It will make the cell even more negative. The potassium will leak out from the leaky channels as well, and the electronegativity inside the cell is minus 70 millivolt. Okay, and the sodium present in the lumen of the proximal convoluted tubule is in high concentration. It will bind to a co-transport protein. The co-transport protein will pull the sodium from high concentration to low concentration, because the electrochemical and the concentration gradient both favor this process. Concentration gradient, because the sodium is already high in the lumen and the sodium is low in the cell, and electronegativity inside the cell, so it means the electronegative gradient and the concentration gradient, they both causes the sodium to move from the lumen into the cell, so it's in our favor, right? But these transporters can also transport other substances along with the sodium. It's known as sodium co-transport mechanism. First is the glucose and second is the amino acid. Glucose and amino acids are low in the lumen and they are high in the cell. They will move against their concentration gradient with the help of active transport, but it's the secondary active transport because ATP is not being utilized here directly. ATP has been utilized in this pump, the sodium potassium ATPases, but it's not involved here directly, so it's the secondary active transport. And thus glucose and amino acid will move from the cell to the interstitium by the facilitated diffusion, because this process involves just moving the glucose and amino acid out from high to low concentration, but they will involve channel protein, so they will called as the facilitated diffusion. And this glucose and amino acid present in the interstitium will be taken up by the peritubular capillaries. because afferent arteriole divides into the glomerular capillaries, then efferent arterioles divides to peritubular capillaries, and they will eventually surround the tubules of the nephrons to reabsorb the material. So we will discuss other substances in this series of lectures. So this was for today, and I will see you in the next video. Until then, goodbye.